Good morning, everyone. I'd like to start our session. I'm Lori Isom, and I'm professor and chair of pharmacology at the University of Michigan Medical School, and I'm also a professor of neurology. And I am privileged to chair the scientific advisory board of the Dravet Syndrome Foundation. I spoke actually at the very first PAME meeting in 2012, and it influenced me so much to go back to Ann Arbor and change the focus of the work in my lab to focus on SUDEP. And, the rest is history. And I, in fact, love to bring my trainees to this meeting because it refocuses their work and it informs the why of why we're doing the important neurophysiological research that we do, and we're developing and working on animal models, because sometimes you can get way down to the weeds. And of course we do, right, in the laboratory, but it's really good to come back here every year and understand what our goal is. And our goal is that there are patients waiting. And so that's what we're trying to do. So it's just my honor today to introduce this really wonderful session and thank the organizers for asking me to moderate. So today we have three wonderful speakers. And what we'll do is not take questions in between, but wait for the panel at the end. So it's my privilege to introduce our first speaker, Vikram Bagchi, who is an MD PhD fellow at the University of Michigan Medical School and a member of our medical scientist training program that is an NIH-funded program. So Vikram is going to talk to us about the development of our rabbit model of Dravet syndrome, which we think is a really appropriate model for studying the neurophysiological basis of SUDEP. And Vikram is an aspiring physician. We hope that someday he'll become a neurologist and contribute to curing people with epilepsy. So without further ado, I'd like to introduce Vikram.
Cool. Hi, everyone. Thanks for having me, and really excited to share our research on such an important topic with you all today. So I wanted to start off by providing a little bit of historical perspective and actually introduce a historical fact, which is Patsy Custis, the stepdaughter of our first president, George Washington, suffered from a chronic epilepsy syndrome. And in fact, if you look up the George Washington papers in the Library of Congress, you'd find 50 diary entries pertaining to Patsy, and they often document her seizures. And in one diary entry in particular, on June 19th of 1773, it simply reads, "Patsy died suddenly today at 5 o'clock. And it was noted that briefly before her death, she had experienced a seizure. And together, this represents one of the earliest documented cases of probable SUDEP. And so over the next 200 or so years, SUDEP as a condition, sudden unexpected death in epilepsy, gradually gained recognition. But there was difficulty in identifying this condition. And that changed in 1997 when Dr. Lena Nashev proposed a unified definition for SUDEP. And she identified three core aspects of this condition. First, the death sudden and unexpected. Second, it occurs in the context of an epilepsy syndrome. And third, it's a diagnosis of exclusion. Other causes of death need to be ruled out. And while this definition helped with identifying cases of SUDEP, as we all know, our relative understanding of this condition is still pretty poor compared to other conditions. And a big reason for that is SUDEP is a really difficult, challenging clinical problem to study. And that's due to its core aspects. Number one, it's sudden. The deaths are unwitnessed often. They often occur at night and occur without any warning. And so it's really, really hard to capture any real-time patient data from this. And number two, the deaths are unexpected. With other conditions, we have the benefit of biomarkers, imaging, clinical diagnostics to look out for things. We don't have those with SUDEP right now. So what's the approach for studying a sudden and unexpected problem? Well, a good strategy is perhaps delineated in the Mortimer study. And the Mortimer study was a study published in 2013, and it stands for the Mortality and Epilepsy Monitoring Unit Study. And this was simply a survey of epilepsy monitoring units around the world about SUDEP events that had occurred on their premises. And the result of this survey was 16 definite SUDEP cases were identified. And the key aspect of this is because these events had occurred in an epilepsy monitoring unit, these events were caught on video with EEG. And this allowed for comparison of these 16 SUDEP cases to one another to see if we could figure out any kind of similarities. And what was identified is that SUDEP seems to progress in a rather consistent sequence of events in these patients, where these patients first had a generalized seizure. Shortly after this, they had difficulty with their breathing, first developing rapid breathing and then the breathing stopping. And then about a few minutes later, their heart would stop. And again, in these cases, it showed this consistent sequence of events. And this was also helpful as it gave researchers an idea about which body systems to start taking a closer look at the brain because of the seizures, taking a look at the respiratory system because of the breathing issues, and taking a look at the heart because it's stopping. But in addition to its clinical challenges, SUDEP is also a really difficult problem for researchers to approach. And that's because SUDEP is actually a heterogeneous condition. And what that means is there's actually different kinds of SUDEP. And to illustrate this, let's compare two conditions. So on the left-hand side, we have Dravet syndrome. It's a genetic epilepsy. It's characterized by developmental delays and generalized seizures. And the SUDEP rate in these patients is pretty high. And often it's affecting young kids and adolescents. And the mechanism that's been noted in these patients is usually a seizure event that's then leading to a mixture of respiratory, autonomic, and cardiac dysfunction. In contrast, if we look at temporal lobe epilepsy, this is often an acquired epilepsy, and it's often presenting with focal seizures. And the SUDEP rate in these patients is a bit lower, and when it does occur, it primarily is impacting adults. And the mechanism that's been noted in these patients is a little different, where it's a seizure, but it's mostly leading to respiratory issues, not really involving the heart. And so the main takeaway from this slide is to recognize that with every epilepsy condition, there's a unique SUDEP mechanism most likely occurring. There's a SUDEP implicated in Dravet syndrome and one with temporal lobe epilepsy. And as you can all imagine, there's so many other different kinds of epilepsies out there, and so they could have a unique SUDEP risk as well. So in the ISOM lab, we've decided to focus in on the SUDEP implicated in Dravet syndrome. And there's two key reasons for this. Number one, SUDEP is very prevalent in Dravet syndrome. In a study examining about 180 patients who had passed away with Dravet syndrome, a diagnosis of Dravet syndrome, it was noted that 50% of these patients had passed away from SUDEP. The other reason is that in Dravet syndrome, it's primarily a monogenic disorder. And what that means is that the majority of patients with this condition have variants impacting a single gene, the SCN1A gene that encodes for voltage-gated sodium channel. And as researchers, this involvement of a single gene really simplifies our approach of modeling this in animals, as we only have to target one gene. And we know that this approach works because in our mouse model of Dravet syndrome, with SCN1A haploinsufficiency, we see a phenotype of spontaneous seizures in SUDEP, which matches up with the phenotype seen in humans. So what have we learned from this mouse model? Well, video EEG of SUDEP events in the mice with Dravet syndrome has shown that SUDEP seems to follow a set sequence of events in these mice. With first, there's a seizure that's impacting the brain, and this seems to cause a spreading depression that impacts the brainstem. And the brainstem has a whole host of important circuits, but namely circuits that are controlling the function of the heart, the function of the lungs, and the autonomic nervous system, integrating the two. And these findings are really important because it showed that similar to humans, SUDEP is following a set sequence of events. And this similarity allows us to kind of focus in and ask even more specific questions of this model. So in the ISOM lab, we focused in on the heart, with the main reason being the gene implicated in Dravet syndrome, SCN1A, is expressed in both brain and heart in rodents and humans. And when we take a closer look at our mouse model Dravet syndrome, and specifically at its heart, we find that the heart cells have altered sodium current density and have substrates for arrhythmia. But our interpretation of this model, especially the cardiac phenotypes, is pretty limited. And that's because of intrinsic differences in the cardiac action potential between mice and humans. As you can see, the human cardiac action potential follows a typical square shape versus the mouse, it does not. And this large difference really makes it difficult to evaluate more nuanced differences that could be occurring. And so for this reason, we shifted to a rabbit model of Dravet syndrome. As you can see, it's a much better model of the action potential from humans, as a cardiac action potential from the shape. But rabbits also have some additional advantages. They're a much better model for human vital signs, as you can see by the heart rate and respiratory rate, which leads to more relevant interpretations of variability in these parameters. And the larger size of the rabbit allows for technological advances. One in which we're particularly excited about is we can implant intra-abdominal telemeters in these rabbits, and this allows us to place a lead in their brain and in their heart that gives us simultaneous EKG and EEG signals in a freely moving animal in an untethered system. So with these advantages in mind, a few years ago, under the leadership of Dr. Chunling Chen in the ISOM lab, and in collaboration with the large translational animal core at U of M, we sought to make a rabbit model of Dravet syndrome. And our first approach was use gene editing to delete 14 base pairs with an exon 1 of SCN1A. And the consequence of this mutation was SCN1A haploinsufficiency. And we verified this by looking at the tissue of brain. And on the right-hand side, you can see that wild-type levels of SCN1A transcript are fully present. And in the middle lane, the heterozygous SCN1A, which corresponds to the human genotype, you can see approximately half, although it's not quantified in this figure. And on the left side panel is the homozygous, where you can see it's absent, but this does not really correlate to a human genotype that's found. And then when we take a look at heart, this is a different way of looking at it. When it's quantified, you can see in blue and purple that these are the wild-type levels where SCN1A is fully present. And then in our heterozygous animals, in red and orange, you can see again, SCN1A is about half present. So despite this biochemical evidence, though, of SCN1A haploinsufficiency, we initially didn't see a phenotype in our rabbits. And these rabbits were on the New Zealand white background strain, which is what's used by our core. And these animals didn't have any seizures, they didn't have any clear arrhythmia, and they had no changes in their lifespan. And while initially we were surprised by this, we wondered if this was similar to the mouse models of Dravet syndrome, which exhibit significant background strain dependency. And for this reason, we considered crossing our rabbits with another strain called Dutch belted. And the Dutch belted rabbit model has been used in other studies to model spasms and dyskinesias, movement disorders. And so we wondered maybe on this background, seizures could emerge. And indeed they did, with the F1 generation developing both generalized seizures as well as arrhythmia. And so let's take a closer look at that data. So one of the first experiments that we did in these DS rabbits, which correspond again to the human genotype of heterozygotes for SCN1A, one of the first things we checked were sensitivity to hyperthermia-induced seizures. And the reason we wanted to take a look at this is because in Dravet syndrome, in the first year of life, kids often have febrile seizures. Not all the time, but often. And so we were curious about whether our rabbits shared this phenotype. And so our protocol involves taking wild-type and DS rabbits and gradually heating them to 45 degrees centigrade. And during this protocol, we're observing the rabbits both visually and, because of the telemeters, electrographically for the emergence of any seizures. And what we can see on the left side panel is on the bottom, with a thin rise of the blue line the thin blue line represents a rise in temperature during this experiment. And the large black signal corresponds to the EEG in the rabbit during this protocol. And this is a DS rabbit that's experiencing a seizure at this temperature. And what's important to note is that after doing this experiment, none of our wild-type animals will seize even until we heat them to 45 degrees, whereas all of our DS rabbits will seize at a temperature of 43 degrees or lower. In addition to hyperthermia and sensitive seizures, our rabbits also have spontaneous seizures. And so what's being shown here are a set of 3 still images of spontaneous seizure that was captured in one of our rabbits. And on the left side image, you can see blurring of the rabbit's limbs, indicating a kind of tonic contraction. And in the right 2 panels, you can see the extension of the rabbit's body, indicating together this represents a spontaneous generalized tonic-clonic seizure. In addition, our rabbits also have a cardiac phenotype. And the way we approach this is we isolate the hearts of the rabbit, wild-type and DS, and we apply a pacing stimulus to the heart to see how it responds. And when we apply this pacing stimulus to a wild-type heart, you can see that a regular sinus rhythm, this is a normal rhythm, emerges. But when we apply the same stimulus to a DS heart, you can see the emergence of an arrhythmia, and specifically, this is ventricular tachycardia. And again, importantly, we only see this phenotype emerge when we apply the stimulus to DS hearts. We never see this in the wild types. And lately, we've also been able to start exploring the respiratory activity of these rabbits. And this is in collaboration with Dr. Erica Levitt's lab, which can do these amazing specialized experiments because of the larger rabbit size by specifically examining the phrenic nerve activity of these animals. And the phrenic nerve is the main nerve that innervates the diaphragm. And it's the main, which is a muscle that helps us during inspiration, during breathing in. And what the Levitt lab shows is when you look at the phrenic nerve from a wild type animal, there's two things to appreciate here. Number one, you can see these very clear spikes happening, and this corresponds to when the nerve's firing. And in between each spike, you can see almost no activity. So this indicates a very clear of the phrenic nerve firing at a regular rhythm. Now, when you look at the phrenic nerve from a Dravet syndrome rabbit, you can see some clear differences. When you look at the spike, it's wider and it's blunted. And in between these spikes now, it's no longer a clear signal. We're seeing these bursts of irregular activity. And so to us, this indicates there's some altered respiratory activity also being found in our rabbits as well. And so I'd like to conclude by discussing the raw seizure tracings we can get from our intra-abdominal telemeters. And the first thing I want to point out is the amount of data that we're getting from these telemeters. At the very top, you can see the EEG signal, and this is a signal, again, from the lead in the brain. And before the seizure starts, you can appreciate very low-grade electrical activity being measured. But after the red arrow and after the onset of the seizure, you can see very clear epileptic discharges happening. And so this is, again, a good confirmation for us visually of what's going on by looking at what's going on electrographically. The second parameter is temperature. And again, it's important to note that in Dravet syndrome, not every seizure is associated with a clear change in temperature. So we're not too surprised that we're not seeing a major change in temperature with this documented seizure. But it's an important parameter that we're keeping track of. The third is accelerometer, and this is basically a movement output for the animal. And again, this is a further control for us, where visually we're seeing the seizure, we're seeing the electrographic evidence of the seizure, and we're also seeing the movement output as well that's correlating with the epileptic discharges. And then the last is the EKG, the signal from the heart. And we're getting to see what the heart does before, during, and after a seizure. And the reason why I wanted to conclude with this piece of data is because one of our hopes is if we can capture a SUDEP event with one of these telemeters running, then like the Mortimer study and with the video EEG studies of the mouse models of SUDEP, we might be able to get a much clearer idea of the sequence of events that's happening in our rabbits, and again, be able to ask more specific questions following from that. So to summarize, I pointed out two challenges with SUDEP. Number one is how do we approach and learn from a sudden and unexpected condition? And a strategy that's been delineated is video EEG monitoring is a great first start. It's helping us understand the sequence of events that's happening in a relatively unknown condition. And the second is how do we model, as researchers, a complex and heterogeneous condition? And a strategy that we're approaching is studying SUDEP in more specific contexts and to try to get really focused in on the mechanisms of disease. And so we've united these two strategies together with our rabbit model of Dravet syndrome with the implanted telemeters. And our hope is to use this model to really study this model and uncover its phenotypes and then ask even more specific questions from that. And with that, I'd like to conclude and thank Dr. Lori Isom for all her mentorship and guidance, the rabbit team in the ISOM lab and the rest of the ISOM lab as well, and then our amazing collaborators without whom we couldn't do any of this work. And lastly, the core with the rabbit model help, and then our generous funding. Thank you.
Thanks, Vikram. Our second speaker is also from the University of Michigan, Dr. Colleen Carpenter, who is currently assistant professor at the University of Richmond. And she's in the department, she's an assistant professor of biology and a Costin Family Fellow in molecular biology at her institution. She earned her bachelor's degree in chemistry from Wesleyan University and her PhD in pharmacology from the University of Michigan. We're so proud of her. And then she completed a postdoctoral fellowship training with Dr. Scott Baraban at the University of California, San Francisco. So Dr. Carpenter is really focused on a different animal model of epilepsy in SUDEP, and that is zebrafish. And I think it's important to realize that, but the definition of model is that it's wrong, but all models are useful. And the truth about the mechanism of SUDEP is the intersection of all these diverse models. Because as Vikram pointed out, each kind of epilepsy may have a slightly different mechanism of SUDEP. And so Colleen is going to talk to us today about her beautiful work using zebrafish that are really an attractive and cost-effective model for drug discovery and disease mechanisms. And her work focuses on using zebrafish to model rare epilepsies, including Lennox-Gastaut syndrome, with the aim of discovering new and effective treatments for these disorders. Colleen, welcome.
Hi, everyone. I'll start out by saying, Lori told me before coming to PAME that this is going to be a meeting that would change my life. And wow, it's been inspiring to listen to everybody, and for everyone that shared, thank you. And thank you for this space to share my work. It's really an honor for me to be here. So I'm Colleen Carpenter. I'm at the University of Richmond, and I describe myself as a zebrafish enthusiast in the sense that I use zebrafish to model brain disorders with a particular focus on epilepsy. We get very biased in science that we're like, our model is the best model. So I'm happy Lori shared that, the hope is that we could use these different model systems to complement each other to answer crucial questions with respect to, brain disorders such as epilepsy. And I wanted to kind of give a, historical view of different models. So traditionally, we've seen the use of rodents, monkeys, different mammals that have been very useful in us understanding the causes of different neurological diseases. And when we think of models, we should include humans as well, because we do test things that we find preclinically in humans in a small population before we kind of put it out into the larger population. We want to make sure it's safe and effective. So these models have been great. One of the things is that they can be very costly. Maintenance can be pretty significant. And for me, I've thought a lot about kind of what we call simpler organisms that we can use to answer critical questions in epilepsy. This ranges from, you could think of bacteria, worms, fruit flies, and my model of focus, zebrafish, and how, although we put them in this group of more evolutionarily distant from humans, they do share similar genetics. Molecular pathways are conserved in these systems. And I want to first start by showing you the benefits of the zebrafish and why I employ it in my research. So this is kind of a timeline of the emergence of the zebrafish as a valid model, a valid vertebrate model, to now where we're employing it for many disorders. So in the 80s is when folks were like, okay, this thing can be respected. We could use it to study different diseases, all the way to the establishment of ZERC, which is a great facility for storing and distributing different zebrafish models that are made, all the way to the first drug screen in 2004. You'll see closely after that the emergence of the first chemically induced zebrafish model from the Baraban lab, from Scott's Baraban lab, all the way to moving from the preclinical work in fish to taking that to the clinic. That happened in 2011. Right after that, we saw the emergence of the genetic zebrafish model of Dravet syndrome, which you've gotten a background to, and that was also from the Baraban lab. And now the fish, or zebrafish, have really served as a great model for us to, again, delve into these deep questions of what is causing different diseases. And with epilepsy, what are these causative drivers? So why do I use zebrafish? So I mentioned genetically they're similar. They share about 70% their genetic makeup with humans. When we think of disease-causing genes, that number pops up to about 85%. They have similar brain structures. So I wanted to just briefly highlight some circuits that you've probably heard about: a dopaminergic circuit, serotonergic circuit. Those are conserved between fish, rodent, and humans. Zebrafish are fertilized externally, so we can access them to study our different questions from time zero. A female zebrafish, adult zebrafish, can lay up to 200 embryos in one go. So when we think of sample size and wanting to test question with larger ends or larger sample sizes, zebrafish are going to provide you with that capability. They develop rapidly by about 5 DPF, or 5 days post-fertilization. At their larval stage, we see a pretty complex brain. They have very intricate motor function, and they can model epilepsy even at this early stage. With respect to the translational aspect, I'm a trained pharmacologist. I think a lot about, how can we get to effective treatments faster, cheaper. I think of the benefits of zebrafish in creating genetic models. There's an ease of drug administration, particularly at the early larval stage. You can dose fish through water. So this is showing zebrafish in a 96-well plate. So for folks that aren't used to that, imagine the palm of your hand is about the size of the plate. And we're studying here 200 fish all at the same time. So we get that level of throughput where we can screen many of these fish all in one go and pretty cheaply. So, with my training at Michigan, we've delved into kind of that costliness of the drug discovery pipeline and how at a preclinical level, it can be costly as well. And that's what's contributing to that high value you see there of $3 billion to get a drug to market. So I firmly believe that zebrafish provide us with a low cost and a high throughput platform for us to get things from the preclinical level into the clinic and hopefully ultimately to patients. One of the things that's very attractive to me is the fact that they're transparent. So at really, really early stages, you could do in vivo studies, tracking, so live tracking of brain activity in these fish. Epilepsy is a brain disorder. So we can really get to those questions of what's going wrong, what's abnormal in these circuits when we're studying an epileptic brain. I know I don't have to describe epilepsy to this audience, but I do want to reinforce certain things that have been said before where, we know it's a chronic disorder where there are spontaneous recurrent seizures. There are many disorders associated with epilepsy. So from the neurodevelopmental issues, sleep disorders, which is mentioned earlier, anxiety, and this session focuses on SUDEP. These are the things that we have to think about when we're thinking about treating epilepsy. It's not just about the seizures themselves, but the complexity of the disease when it comes to the clinic. We need to address all these different ailments. So there are 1 in 26 persons in the US that will develop epilepsy. Currently, about a third of those patients do not respond to anti-epileptic treatments. And, we've been hearing the message of, having these uncontrolled events can lead to many other issues, including SUDEP. So there is an urgent need for us to make new treatments without bad side effects, right? And I think our zebrafish models can help with that and hopefully reduce the economic costs to the population of folks suffering from epilepsy and for the caregivers as well. So with the evolution of the field, we've come to appreciate more the genetic drivers of epilepsy. This list is a tiny list compared to all we know now with the different genes we think that could contribute to the different forms of epilepsy we see in the clinic. This is from genome sequencing of patients with epilepsy. If you scan through, depending on your awareness of genetic epilepsies, you're probably like, oh, I know this one. We talked about Dravet syndrome and the link to SCN1A. I'll mention that model, STX-BP model, BP1 model, which is linked to Lennox-Gastaut syndrome. But with my postdoctoral fellowship in the Baraban lab, we looked at the validity of modeling different genetic epilepsies in zebrafish. If we can recreate mutations that are found in patients in these fish, do we see the complexity of kind of phenotypes or symptoms that we see in patients in this fish or in these fish? The goal here is to understand that, are these just correlations or are these causations? Are these driving the epilepsies? And once we have valid models, can we then use that for drug screening to see if we can find new therapies for these epilepsies? So when I got to the Baraban lab, we have this very, I will say, when I joined the lab, I was like, okay, so we're really doing this. So there are all these genes, we're gonna model them, we're gonna screen all these little fish, and we're gonna see, their predictive validity with respect to epilepsy. A lot of work went into this, a lot of people went into this, and I just wanted to highlight some of the work I did there and then what I've been doing in my lab currently at the University of Richmond. So here we have recreated genetic mutations, loss of function genetic mutations that we find in patients in zebrafish. And then with modeling epilepsy, one of the things we're looking for is, do they have spontaneous electrical events, abnormal brain events that look like seizures? Do they have behavioral seizures? Do they have other phenotypes such as SUDEP that we associate with epilepsy? So here I'm highlighting, looking at larval survival. So do we see increased cases of premature death in these zebrafish? So I've ranked, so this is a heat map to kind of orient you to this figure. So just think that the more red you see is the occurrence of spontaneous seizures. So we find that in mutating, so doing monogenic mutations in fish, that there are some that emerge with robust seizure activity. So they spontaneously seize, and they also have other, what we think are symptoms or phenotypes that match comorbidities that we see in patients. So I wanted to start out with the Dravet model. So that had been, that data had been out for a while, that these fish spontaneously seize before this epilepsy zebrafish project. But using CRISPR to make these new line of fish, we wanted to use CRISPR to recreate our Dravet model to make sure that we could reproduce that data before looking at the other fish models we made. And similarly, as we had seen before, these fish spontaneously seize. This is looking at, I guess my cursor's not working, seizure events in these fish at different time points. So DPF being days post-fertilization. So by 4 to 5 days old, you see spontaneous seizures in these fish. They prematurely die. You start seeing death even as early as 8 to 9 days. By 12, most of these fish do die, and we think that's a marker of SUDEP. There's a question earlier, I don't know if the person's in this audience. If you want to ask more questions later, you're welcome to. We find that serotonin modulating drugs, including clemizole, so clemizole was first put out as an antihistamine, but it was found in the screen to block seizure activity in Dravet zebrafish, and other serotonin modulating compounds also do the same thing. So one of my questions was, if we're modeling seizures and this is blocking those seizure activities, what about the comorbidities, and do we see some effectiveness with these serotonin modulating compounds against those phenotypes as well? So with respect to other comorbidities, so we do different assays to monitor kind of different, we'd say, symptoms of epilepsy in these zebrafish. So one assay I've done is a startle response. So you can imagine if we're all in this room and it's kind of dark right now, and if you all of a sudden turn on the light really, really bright, you'll probably have, you'll be triggered, right? And you're like, what's going on? Should I leave? What's happening? The same thing happens with fish. They have a sensory motor integration where you sense the stimuli. You normally have a motor response, it's an escape response. Once you realize that that stimuli isn't harmful, you what we call is habituate to that signal, so you stop producing that response. Here we find wild-type fish, so they have a response to the stimuli, which habituates over time. In our Dravet fish, we actually see a hyper response. So there's some abnormality in the stimuli detection to that motor response, and we think that's a marker of neurodevelopmental issues. We also see that phenotype in one of our models of another rare DEE. So pyridoxine-dependent epilepsy is modeled by ALD87A1 knockout zebrafish, and we also see that phenotype as well, and we do see it in some of our other zebrafish lines. We also have, or I did an assay looking at anxiety-like phenotypes. So normally zebrafish at really early stages have a light preference. As they get older and more pigmented, it shifts to a dark preference because they're hiding from predators. That's been thought to be the evolutionary purpose of that. So we find with these mutant zebrafish is that they have an altered perception of kind of safety. So they actually run. I remember doing these experiments. You put them in the plate, they like run to the dark, and then they just stick in the corner. And we think this is we call it stigmotaxis, which is wall hugging, a sign of anxiety. So we see this, some of my things got lost, so we see this quite robustly in these mutant zebrafish. And I wanted to go back to the story of clemizole, where we saw that it blocked seizure activities. We also seeing it blocked, or we saw it block some of these comorbid phenotypes. So here I'm actually showing you a heat map representation of that startle stimuli data. And we've given clemizole, valproic acid, and stiripentol. So those are common anti-seizure drugs. Those actually don't work as well in blocking this startle phenotype in this Dravet fish compared to clemizole. And you could see it's less red or less pink. And that's because it's actually bringing it down to wild type levels. So we're seeing this effectiveness of these serotonin drugs for what we think are comorbid phenotypes. We also saw that with anxiety as well, that giving clemizole acutely increased the chance of these mutant zebrafish exploring, so not just sticking to the wall, but actually exploring the arena, so increasing their exploratory behavior. I mentioned we modeled a lot of different genetic epilepsies in fish. I won't show you all that data. One that I'm actively working on is STXBP-related disorders in our STXBP1B knockout zebrafish. We also find that these fish spontaneously seize. They live a little longer, but they do die within 3 weeks. So we, again, think this can be a marker of SUDEP. Interestingly, clemizole also works to reduce seizures in these fish, and other serotonin modulating drugs do this as well. And with respect to comorbidities, we find, and I'm only showing you one data set, this is looking at our startle response. We actually see a reduced startle response. They really don't respond to changes in light. We also did other stimuli as well. We've seen social deficits, abnormal exploratory behaviors. And again, we are having all these pieces that we think are matching the complexity of epilepsies that we do see in patients. So finally, I wanted to highlight one thing that's been conserved in looking at these different genetic models of epilepsy. So I pointed out our Dravet model, our STXBP1 model, but we've screened many others, some having spontaneous seizures, but what we've seen as a common trait is this premature death that we think is a good marker of SUDEP in these models. So I think this, to me, gives me confidence that this could be a great system for us to further tease apart the mechanisms that are driving SUDEP, and with zebrafish and their translational potential, we could use this as a great way to screen for novel therapeutics. So to kind of recap, so with my work, I've been really focused on the genetic drivers of epilepsy and SUDEP. I think zebrafish are a great drug screening model and a good mechanistic model as well. Like, we could really think about what's going on to lead to SUDEP, for instance. There's ways, so I don't do this work currently, but I'm very interested in cardiovascular function in these models. And this is something that there are tools for that are available. I will end with thinking about limitations of the models. This is why I think all the models are important. There's things we can't do as well with fish. If you could think about a fish swimming around, like there's not everything that we can model. But one thing I wanted to point out is that we actually did some preliminary work looking at clemizole and SUDEP, and we didn't see a reduction in the cases of premature death with giving clemizole. But I mentioned before that with dosing fish, we've used the strategy of just putting the drug in water, it gets into the fish. There's dosing kind of limitations when we work with fish, and I think there are better ways that we can do this study to mimic maybe a more chronic administration of drug to look at whether that's necessary to augment this premature death phenotype. With the models we've made, it was easier to do monogenic models, but we know, or with the field, we think that in many cases, epilepsies and cases of SUDEP might not be from one gene mutation. There could be a couple different things that are contributing to the presence of these phenotypes. So with the models we have now, I think we don't see some kind of phenotypes because they're only monogenic. So I think work needs to be put in to make, double knockouts or triple knockouts to see if that's when we can get true replicas of, again, the clinical manifestations of this disorder. To end, I wanted to I should start by thanking University of Michigan, gave me a strong pharmacology background. But with respect to the data that was produced that I've shown you here, other people from the Baraban Lab contributed to some of the work I presented. My current research group is all undergrads. I work at a predominantly undergrad university. I wanted to be in this environment because I remember how inspiring it was when I was an undergrad researcher thinking that I could contribute to science. And I actually have had a lot of students reach out because of their personal connection to epilepsy, and it's been a great way to provide them with an environment where they could learn more about the disorder and practice it with respect to practice science that could contribute to the understanding and hopefully better treatment of epilepsies in the future. I'm funded by the STX-BP1 Foundation currently, and my startup from University of Richmond. And I just wanted to thank the PAME organizing committee. This has been a great meeting so far. And again, thank you for this space to share my work.
Thank you, Colleen. That was really inspiring. Great work. So our final speaker is Dr. Laura Lubbers, who is Chief Scientific Officer at Cure Epilepsy. And in her job, she's responsible for developing and executing Cure's research strategy and all research programs that seek to cure epilepsy and not just treat the symptoms. And Laura earned her bachelor's in physiology, her master's degree, and her PhD from the University of Illinois, and came to Cure. She also has a personal story and a personal interest in epilepsy, and that her sister Ellen suffered from intractable epilepsy from an early age, and then her mother was diagnosed with epilepsy later in life. So she's all in to cure epilepsy. We're so glad she's here today, and she's going to talk about the use of common data elements in our research with animal models so that we can all communicate with each other. So Laura, thank you.
Thank you so much, Lori, and thank you to all. Yes, I am all in, but the thing that inspires me so much about this community is that I know that you are all in too. There is no more passionate community doing research and I've traveled around in my research experiences, so I just want to thank you all for the passion that you bring and for our the previous speakers and the models that you're working on, truly inspiring, and really helps set this up well, I think. We have some beautiful examples of how we can really translate basic research knowledge to the human condition. And again, I applaud everybody in the audience who is really working on doing just that. And even in the plenary session, hearing Rama talk about the sleep modeling that's going on and that clear desire to translate basic research to the human condition. And that's really what this is about. We've heard about these models. We know that this is a desire within our community, but as science is getting more and more complex and the model systems are being more complex, we really need a good way to communicate what it is we're doing. And so this talk on common data elements is intended to do that. So I'm going to tell you why standardizing research using common data elements is important for the SUDEP research community. I'm going to tell you what Cure Epilepsy did to contribute to the solution of data standardization, and what you can all do to continue to help progress this area and implement these tools that we created. So the story I'm going to tell you in this project that I'm going to talk about was really inspired by the SUDEP Summit that Lori was very involved in, and I know many in the audience were very involved with. That was hosted by the Epilepsy Foundation in 2020. That summit brought together an array of stakeholders to talk about key issues within the SUDEP community as a way to prevent SUDEP. And also, brought in basic researchers to really talk about what are the things that we need as a community to continue to advance this area. And as the discussions proceeded, there were a number of topics where there were some clear themes, and reading the summaries where basic researchers really wanted to be able to align their basic SUDEP research models with clinical experience. They wanted to have a standardized reporting for all SUDEP models and methodologies. They had a long vision for a searchable, evolving database with all epilepsy-related models, and they really wanted to bring in the clinical component to that as well. And they recognized the need to have community or committee oversight to be able to evolve this over time. But how in the world do we do that in this very complex environment of science that we live in? How can we align the data to achieve these goals? And this is a first step in doing that. So as a community, we can start by standardizing the ways we report the experimental data that are developed within our studies by employing common data elements, or CDEs. I'm sure I'll be slipping into saying CDEs, even though I dislike acronyms. Common data elements are a means of describing the details of a research study in a consistent way so that others can clearly understand those details and the outcomes of the studies. This would seem to be commonplace, but again, as science is expanding, we lose track of a lot of those details, but they are essential. We know time and time again, people try to repeat something in the literature and it fails, and we don't know why. So having common data elements in place can help with that. The common data elements are really basic building blocks of research studies. And we've already had some great examples provided to us. And I think Colleen and Vikram set this up beautifully for me, and I probably don't have to, belabor this. But certainly if a researcher wants to study a cardiac endpoint, there are lots of options to study in a mouse model. A researcher may say, well, I want to use electrocardiogram measures to measure a variety of things. To measure heart rate variability or QT interval or any number of other endpoints that one can extract from this simple measure of cardiac function in a rodent model. As we dig in deeper, we know as scientists, well, there are even smaller units of information there. There's the beat to beat variability. Are we studying that? Are we studying just reporting out means? How are we really communicating our science? And how do we do that when we incorporate other species? How do we have a common language across these species? Again, it's getting even more complex, but we all know, and I know, it has come up many times already in this conference about the need to translate to the human condition. So common data elements serve researchers in the following ways. They serve as a guide for the important research endpoints to be used and captured in a study. They provide clear definitions and instructions for use so that researchers can collect data the same way in every study. They ensure that data collected the same way across multiple studies. They increase the transparency and reproducibility of data being collected, and they help ensure data harmonization, should that be feasible. And again, that's that longer range view into, someday having a database and being able to compare studies. We know that SUDEP is rare. We know that we have to bring as much data together to truly understand the biological causes of SUDEP. So CDEs can help us in these ways. So Cure Epilepsy set about tackling the initial steps of this challenge by bringing together so many of you in this room, developing a steering committee and working groups with specific expertise to be able to speak to the different components of SUDEP research studies. It was important to gather both preclinical or basic research scientists as well as those with clinical expertise to make sure that we were really developing these translatable common data elements that could be used and ensure that multiple perspectives were included. In doing this, we identified and defined over 100 CDEs relevant to SUDEP research, and we assembled them into what we call case report forms, or CRFs. An example of a CRF is shown here. I hope it's big enough on the slide. And it's a very simple form. Many of you will not be surprised by this. It kind of looks like your lab notebook, but it does help define more specifically those elements that are important in SUDEP research. So in addition to this image that this core module that I'm sharing with you, and again, you can fill in the details of the laboratory environment, the details of the experiment, including the animal model, the strain. If you're using rodents, we have many strains to work with, and other factors to consider, like age and gender, or sex, sorry. So we have that basic core module, and we also developed 6 other modules that may relate to specific endpoints within a researcher's study. This allows researchers to really have that core set of endpoints, but also to be able to have other other tools that speak to perhaps the physiologic measures, like the cardiac and the respiratory measures, and specific data elements within those physiological functions. We have one for therapies and pharmacology to be able to record those and recording the components that go into using and testing drugs. So there are a number of different tools that have been developed as a part of this process. We also developed what are called data dictionaries. And these help researchers clearly describe the data that are being collected within a study. This is a bit of a complicated slide, but it really speaks to just the factors that are captured within those data dictionaries. Again, include just the common data elements that have been described, definitions that have been described for those common data elements, various units of measure that can be considered, and permissible values, yes, no kinds of answers. These data dictionaries can be used to convert these common data elements into electronic formats, Excel spreadsheets with drop downs, as well as hopefully REDCap resources as well. So ultimately, having all of this described could help researchers understand how studies are similar or different, and perhaps describe why results line up or do not. So we hope that these tools will be helpful. What you can do is learn more about these common data elements. We have a section on our website, cureepilepsy.org, that's specific for researchers and provides these tools for you to use. We have 2 publications that are in progress and nearing completion. I hope you will refer to those in using these SUDEP CDEs. I also want to point you to the NIH website, specifically the National Institutes of Neurological Disorders and Stroke Common Data Elements website, which really goes into details around using common data elements. The NINDS has invested a tremendous amount of time and energy in building out common data elements for epilepsy. SUDEP was a gap, and we found that gap and we filled that gap. But you can certainly go to the NINDS website to fill out more, to gather more information and learn how to use them. You can start to use them in your work. The QR code on the slide will take you directly to our website, and to the red features in the slide. Those are all hyperlinked. You can use those to click on it and download forms that have been created. We still have to polish them up a bit, but the basics are there. And you can select what will be useful for your research. And the most important thing that you can do is use these CDEs, reference these CDEs in your publications so that others really can understand your work. The example that I've provided here actually does not use the SUDEP CDEs because we're just releasing those, but this publication by Christos Laskaros and Helen Scharfman really does a nice job of simply laying out how they've used incorporated CDEs and reported them in supplementary components to their publication. So it's just a great example to look at, and it's pretty simple. So again, I hope that these tools will be helpful. Again, I think it fulfills some of the needs that were called out in the SUDEP Summit to be able to align SUDEP research, incorporating those critical clinical components in our thinking, standardizing our reporting, and then again, having oversight through this amazing community. And ultimately, we hope that using these tools will increase the transparency of the methods and the data in your studies. These are really the building blocks that are essential to know. We hope that it will help align the data across research studies and ultimately help understand the biological processes and underlying SUDEP in animal models and humans so that we can find better prevention strategies as a whole. So I want to thank our steering committee. I know, again, many of you are in the room, as well as our working group members. I hope that you are already taking the charge of using these in your labs. And I want to thank the audience in general. If you have any questions, I'm happy to answer them. You can go to our website for more information. Thank you.
Stay with us here. So thank you so much, Laura. And I'd like to invite our speakers to come to the podium. Do we know yet why this seizure on this day? No. Do we have a single animal model of SUDEP? No, but I think that's important because we need many models communicated in the right way and sharing data in the right way to inform each other, because there's not a single mechanism of this very, very complex perfect storm that is SUDEP. Are we there yet? I think we're gonna discuss that now, and I'm looking forward to everyone's questions. So, thank you. Let's get started. Do we have microphones? Yes. Okay, there's Anne back there. So please raise your hand.
Hi, back here, sorry. Okay, can you hear me now? Laura, thank you very much for your presentation. I had a question on what you would envision if somebody is developing a new technique and maybe it involved several of those case report forms and brought in CDEs from several different areas. What do you think is the best way to integrate all of that? Do you want to keep them separate, make your own? And then, then if you go to share it, you're sort of sharing something new that you've kind of made that's tailored to your project specifically.
Yeah. Well, I think, and hopefully, is it on? Yeah, okay. I think these tools can be modified for use, to an investigator's study design. It's not intended to be totally prescriptive. You have flexibility to use these tools. We just want you to use something. And then using the data dictionaries helps provide descriptions of the data. And this is going to be evolving. Science is evolving all the time. I hope that we can come together as a community in years to come to look at the new models. And I've already told Colleen that I'm going to tap her for CDEs for the zebrafish, and there will be new models. So this will be an evolving process. It's just important to be able to communicate clearly. So if it makes sense to have separate forms, I think that's great. If you want to combine them, you can look at Christos's examples as well and just set up the descriptors in a similar way. It's really intended to be helpful.
Hello, thank you for the talk. All of your presentations were really informative, and my question is more towards the cardiac idea, what Dr. Vikram presented. So my idea is, what do we know about whether if we can, if there is, no seizure as a symptom, but can there be SUDEP that is existing? So this, again, I'm asking this because there are patients who have motor impairments but not seizures. If in SCN8A or SCN1A epilepsies or movement disorders, they have movement disorder, but they do not manifest seizures. So my question is, would there be a possibility that the brainstem, the neurons or the interneurons that are part of the brainstem can have, for suppose, an SCN8A mutation, and that's only driving SUDEP through the cardiac arrest or other pathways, but not actually manifesting a seizure before the seizure can be manifested.
You want to take that?
Yeah.
Okay.
You guys hear me? Oh, yes, you can. I think that's a great question. With regards to, I think with regards to SUDEP, the main thought is that a seizure is kind of the first event that's going on, but you bring up a really good point of we don't really know exactly what's going on in the brainstem, but it does seem to be very important in what's going on here. And so I think the question that we're really curious about and focusing on what you're saying is exactly what is the brainstem's role, as it's kind of means the main relay center where it's integrating all these different circuits. And so I think the honest answer to your question is we're not exactly sure at this point, but it is something that we really want to focus on because it's such an important relay center.
Yeah, and I think I'd like to add to that. And in our animals, we see central apnea in the first 2 weeks of life. In our model, we see the development of cardiac abnormalities before we see the first spontaneous seizure. The earliest spontaneous seizure we've seen in our model is about 60 days, okay? But before that, there's all kinds of intrinsic cardiac electrophysiological differences, right? And the earliest difference is that apnea, right? And the dysfunction of the brainstem, the respiratory centers in the first 2 weeks. So if that really translates, that's an important biomarker. Yeah.
So for starters, congratulations. It was a wonderful session. And thanks for putting it together, Lori. Two questions. The first is driven by one of the early slides where the rabbit didn't die. Had the mutation, rabbit didn't die. Highlighting, and you said it, the genetic background is so important. I think Candace sort of got to it at the end. Do we need triple knockouts? But given the reality that background is so important, what do we make of all these models which may or may not have any relevance to people? Now, we're judging it by they do die and they do have electrical discharges, but that doesn't mean it's the same as in people. So the first question is, how do we think about genetic background? The second question, and Lori knew I was going to ask this, is from your work in animals, are there observations or experiments that can be done in people with Dravet syndrome that would validate the mechanism seen in a model system?
Yes, I knew you were going to ask that, Chuck, so thank you very much. So we're working with Julie Zilbro at Michigan, right, who has seen, she is leading the Stoke trial at Michigan, right? And she's working with all these little kids with Dravet. And so I think the apnea issues are something we can actually measure early in the clinic and see if that's true. And I think I'm really excited about that. So is she. So going forward, that could be true. So if you think about the genetic background, that's true in mice too, right? I mean, Jennifer Carney made her model on the 129 background, and you don't get any Dravet phenotype until you do the F1 cross with C57 black 6J, just like us, very similar, okay? I think if you think about, Dravet is usually de novo with SCN1A, but there are families that rare, where they're inherited. And some of the people in the family have no symptoms, some of them have GEFS plus, and some of them have Dravet. And so doing the deep sequencing on those individuals is really, really important. That's where you're going to learn about the genetic background. And I know those studies are ongoing. What we're also doing in collaboration with Gemma Carville is the deep sequencing of our rabbit model, right? The two backgrounds in the Dravet, so we can figure out what those modifiers are, right, in a more physiologically relevant model. Okay.
Can I add a quick point? I think is this working? Yeah. Sorry if I'm looking down. This light is really bright. But with respect to animal models, I think a huge question is predictive validity. And we do have to, in cases where we have clinical data, for instance, some drugs that work somewhat and some drugs that don't work at all, we could evaluate that in our different models. And I can give evidence of the zebrafish with Dravet syndrome. What we find more effective in patients is what we do see as more effective in fish. So I think there's different ways that we can extract from what we do know to kind of serve as a way to say, like, okay, will this actually translate? The new things that we find, will that translate in the end?
Vikram, that was an excellent talk. Thank you for walking us through the history and the development of the rabbit. You know, I'm obviously a little biased on it there, but can you help us understand, why are these, mutant rabbits developing these arrhythmias? Why were you able to more easily achieve these pacing-induced arrhythmias? And as Lori mentioned, you've done some electrophysiological characterization. Does any of those results support a potential mechanism for why they're developing arrhythmias? And then while I have the microphone, Laura, quick question for you, but it's a huge answer. How are we going to get buy-in with the CDs?
So to answer your question with what's going on with the heart, so to remind you all, we focused in on the heart because the main gene implicated in Dravet syndrome, SCN1A, is expressed, and its protein product, the NAV1.1 channel, is expressed in both heart and brain. And when we've taken a closer look at the heart, we've seen that the sodium current density, which is what NAV1.1 is, a sodium channel, in mice and in mice with the SCN1A haploinsufficiency, there's an adjustment in the sodium channels. And we can see that in the heart tissue, in the cardiomyocytes, we're seeing this change. And when we record from this electrophysiologically, we're seeing delayed after depolarizations, which are substrates for arrhythmia. And so the idea is that the gene SCN1A, as an encoding for an ion channel, and the compensation that's happening in the body is, especially in the heart where SCN1A is very much expressed, that's why it's implicating this arrhythmia. What was the second question?
What currents are different?
So, and Laura, you might want to back me up on that or correct me on, but it should be increased NAV1.5. And so that's something that, again, we're trying to parse through, but in the mouse model, it's hard to do that because the inherent differences of that cardiac potential are just so different that really getting at the nuance is difficult.
Yeah, there's that classic in biology paper, Auerbach and Isom et al., right? The first that showed, right, increased sodium current in the mouse model of Dravet, and Roberto finds that again in the rabbit. So there's increased sodium current. There's differences in the family of potassium currents and calcium currents. Okay, yeah.
And to your question about buy-in for the CDEs, would love to hear, I know that there are folks in the audience who are using the CDEs, so I'd love to hear your experiences with them already. But I think the buy-in really comes in seeing these as valued, as an important component of reporting out on your research, and that these are intended to be helpful for you to do that in a way that's consistent with, again, translatability. There are some other factors at play as well that I think will influence buy-in. There are now new criteria about data sharing that are coming through from the federal level. So our hope is that having CDEs available will enhance that data sharing requirement. And I know that our funders, that Cure is a funder, NINDS is certainly interested in also using these CDEs to help define particularly SUDEP models as they're looking at grant applications, as we're looking at grant applications, to try to really enforce that the community-based endpoints are included in those. And so I think, and that helps discern what's a SUDEP model and what's not a SUDEP model. And I think that there's flexibility in there. You have to prove it's a SUDEP model. You can propose endpoints that may be more non-traditional and just establish that. But I think these different factors will help influence buy-in.
I think we have time for one more question.
Hi, how are you? Elaine O'Loughlin, Cure Epilepsy. Excellent talks. I think the complexity of animal models, you need it in your preclinical research and also for translational purposes. And this is directed at you, Colleen. I think of, having that high throughput, having all your animals and your ends in a 96 well plate is huge. And then also, picking out that H1 blocker. And maybe it's more so thinking of my question directed at like fundamental mechanisms or biology in your model. There's some reports that H1 blockers are quite positive in myelin repair in MS with clemastine. I'm wondering, in your model, is this another endpoint that you could read out, and do you see myelin defects in your model and potentially something that you could, look at or repair with the clemastine, the H1 blocker, or is this of interest? And it's very positive to see that, okay, well, they're out there on the market, and could it potentially be something that you could study in your model system?
I would say there's the potential to study it, but I wanted to clarify that the H1 mechanism is actually not conserved for epilepsy. So it was an H1 blocker, but what the thought is that it's a serotonin modulation. So the other H1 blockers don't actually work against Dravet syndrome. So there's ways to tease out, how serotonin is connected, but I want to make sure that the H1 aspect we don't think is relevant, at least for Dravet syndrome. Yeah.
Okay, so let's thank our speakers one more time. Thank you all for your participation, and let's break for lunch. Thank you.